Hi, good afternoon, everyone. My name is Upi Singh. I am privileged to help lead the session on vaccines. So we've had just a great conference so far, heard about everything from public health to data and research, clinical care, and health disparities. And I think this is sort of the next segue in terms of how we can help decrease health disparities and bring a hopeful sign to everyone. In terms of vaccines. So we have a great session for internationally renowned speakers, and I'll just introduce people briefly and then we'll go ahead and start. I do think for all of us, vaccines represent such a hopeful moment. And for me personally, it's three things that I think we have the most thing to be grateful for. One is the science that's helped us get here in an amazing time frame so quickly. For me, the second thing is the patients who participated in all the clinical trials and have, again, helped us get to this point. And the third is all of you, the providers, the health professionals, those in public health who are sort of helping now guide our patients to getting the vaccine. So I look forward to hearing the latest updates from our panelists. I will ask everybody to please stay on time so we can have a really robust discussion at the end. So I'll just briefly introduce people. We'll first have Dr. Dale Hu, who'll come talk to us from NIH. He's back on the farm, so to speak. He did get his undergrad degree at Stanford and then has had numerous leadership roles at the WHO as well as the CDC, and has been at the NIH now for a few decades, where he's led a lot of the work on HIV vaccines as well as other things. And then we'll have Dr. Linky Zhang, who's going to join us from the Comprehensive AIDS Research Center in Tsinghua University in China. Dr. Zhang trained at Edinburgh University and then also at the Aaron Diamonds AIDS Institute in New York University, and has been back leading his program for several years. Then we'll have Dr. Anup Misra, who is an expert in diabetes research. Again, has trained all over the world, including in programs in London as well as UT Southwestern, and is an internationally recognized expert. He's going to talk to us about vaccines and the intersection of diabetes. And then our last speaker is Dr. Aruna Subramanian, who is a professor at Stanford University. She has had training at premier institutions and helped at Johns Hopkins University institute and lead the program for infections and treating infections in immunocompromised hosts. And she also does lead that program at Stanford as well. And so she'll talk to us about vaccines and the immunosuppressed patient. So I think we have a great mix of speakers and talks, and really look forward to hearing the scientific as well as global perspective. So with that, Dale, I'll hand it off to you. Thank you.
Right. Dr. Singh, thank you so much. It's really an honor to be invited to this session, and I really look forward to hearing the other speakers and the discussions. So good morning, good afternoon, and good evening, wherever you are. My name is Dale Hu. I'm the chief of the Vaccine Clinical Research Branch in the Division of AIDS at NIH NIAID. Next slide, please. Before I talk about COVID, I wanted to really start by saying that the traditional vaccine development was a very lengthy, risky, and expensive process, as illustrated by this diagram from Dr. Nicole Lurie's New England Journal of Medicine article from last May. In contrast, the COVID vaccine development was very much accelerated by moving multiple processes in parallel and at risk. In other words, normally the process is very sequential, and what is happening here is some of the processes that occur later have started earlier and in preparation for when the earlier process finishes. Next slide, please. Now, the rapid response in developing a vaccine against the virus, SARS-CoV-2, is really unprecedented historically. Shortly after cases of pneumonia-like virus were first reported in China, the first genome sequence was reported to GenBank by Zhang and colleagues. And within 68 days, so just shortly after 2 months after the sequence selection, there was the first human injection of the mRNA 1273, which was the product used in the Moderna trials. Now, obviously, this rapid response did not occur out of the blue. There were a lot of existing technologies, concepts already existing. There was a very strong vaccine clinical infrastructure evaluating safety, immunogenicity, efficacy, as well as correlates of risk and protection. And the importance of kind of coordinating all these things was really critical in moving things forward. And I want to also mention that in terms of the NIH, there were already a number of existing NIH-supported networks that later formed what was called the CoVPN. And one of the networks that I worked with very closely was the HIV Vaccine Trials Network. And all of this, again, was built on existing infrastructure that was developed over decades. In the next slide, please. Early on, I think scientists looked at a number of existing platforms as illustrated in this table. And obviously, we don't have time to look at each of these, but each of these vaccine platforms has advantages and limitations. And some of them are relatively new, such as the mRNA, and others have a long history, such as live vaccines, protein immunogens, as well as other approaches. Now, clearly, no single vaccine or vaccine platform alone is likely to meet all the global needs across the world. So I think early on, the NIH leadership led by Dr. Fauci was really critical in pushing multiple simultaneous approaches. Next slide, please. Now, this slide illustrates the major vaccine candidates whose clinical efficacy trials were supported by NIH, BARDA, and other federal support. Please note that the Pfizer-BioNTech vaccine is not shown here, even though they did receive support for manufacturing, but not for the actual clinical trials. And the other point that I think bears noticing is that the incredible rapidity in which these trials moved from phase 1, 2, and phase 3 efficacy, and then finally to getting EUA approval or emergency use authorization. The last point I want to make is even though many of these companies had very well-established vaccine programs such as Sanofi Pasteur, there were always risks that happened. In the case of Sanofi, there were manufacturing challenges and protocol delays that resulted in their trials now probably not happening until later this spring. So it shows that really the necessity for having multiple approaches. Next slide, please. I think I want to just summarize now. Obviously, this is a very big field, and I'd be happy to answer more specific questions, but it really illustrates the importance of having existing resources and infrastructure in place to pivot to any emerging pathogen. And even though this was moving at the original name was Operation Warp Speed, there really wasn't any shortcuts in terms of safety. I think through our partners at the FDA and at the CDC, vaccine safety was very meticulously assessed, as illustrated by the rapid reports of any rare side effects that might be associated with vaccines in general. So given the rapid success, I think we do need to keep in mind current and future challenges. One obviously is the emergence of variants. And this is sadly illustrated around the world, most recently in India, but also in South Africa, in Brazil. And I think, you know, we really need to keep alert for these variants of SARS-CoV-2. But SARS-CoV-2 certainly will not be the last emerging pathogen to threaten human societies. There will be other pathogens. And also, as illustrated in the previous session, I think another challenge is the issue of vaccine hesitancy. The success of vaccines has made vaccine preventable diseases seem like they're very rare, and then people sometimes, I think, equate the danger of rare side effects with the danger of vaccines, which I think is really unfortunate. So all of this, I think, points to the importance of expanding public health capacity as well as research so that we're always ready to address any emerging pathogen. Thank you very much.
Thank you so much, Dr. Hu. That was great, a great summary, and I'm sure that we're going to have lots of questions in the open session. So our next speaker is Dr. Zhang, who's going to talk to us about a topic that's obviously a very timely issue, SARS-CoV-2 variants and the impact on antibodies and vaccines. Dr. Zhang?
Hello, can you hear me?
Yes.
Dr. Singh? Yes.
Yes.
This is a great honor to be here and nice to see Dale and others on the same panel. And also a great honor to participate in this very high level discussion. I'm going to talk to you about today is the SARS variants and impact on antibodies and vaccines, primarily from the micro level. So you could see what the virus look like and how that interact with antibodies and what the mutations impact on the serological response to those mutants. I'm Zhang from Tsinghua University in Beijing. In fact, this is a 3:57 o'clock in the morning. It's a very different time zone. This image highlights the real SARS-CoV-2 viruses. And then, in fact, it is a cryo-electron tomography obtained by my colleagues at Tsinghua University, Dr. Sai Li. As you can see, the SARS-CoV-2 look like a blue basket containing so many eggs, and on the surfaces have so many different type of spikes. Some of them are highlighted in red, some highlighted in the light red color. In fact, the blue are the viral membrane that containing the spike proteins. The spikes have different conformational structures. It's very important for the virus latching to the receptor ACE2. At the same time, it's also a major target for neutralizing antibody response and some binding antibody response as well. It is the major antigen that used in developing the vaccine that Dale just mentioned to you. At the center of those yellow eggs are the nuclear capsid that containing the SARS nucleic acid materials. Next, please. So as soon as the outbreak and sequence became available in the database, the scientists around the world are rushing to isolate and characterize many different antibodies against that spike protein. In particular, part of that protein called receptor binding domain here highlights in cyan color. And this domain is critical to bind to the receptor ACE2. So throughout the systematic characterizations, our group and many other groups in the US and in Europe have identified potent neutralizing antibodies that are able to interfere the proper interactions between RBD and ACE2, thereby to prevent this process and prevent the viral infections. The antibodies certainly have a different structure and different functions. Here I listed 3 representative ones. The one on the far left, highlighted in the purple and yellow, is one particular type of monoclonal antibodies. As you can see, it has some spatial clash with the green, and thereby interfering the interaction between RBD and ACE2. The ones in the middle almost overlap completely with ACE2. So you can see the antibody compete with ACE2 in binding to RBD, thereby could block viral infections. The antibody on the right recognize the root part of RBD, and this antibodies does not have potent neutralizing activity. The tools listed on the left, although they recognize different epitope on RBD, both of them have a very strong neutralizing activity. Next, please. However, over the course of the last year, due to the rapid dissemination and transmission of these viruses, SARS-CoV-2 are able to generate an increasing number of variants, here listed, highlighted in red. This really highlights the trimeric structure of the envelope on the spike, although here represented by different color. The yellow represent the RBD portion of the spike, and the CN represent the N-terminal domain of the spike. As you can see, majority of the envelopes, majority of the mutations occurred in both regions. And then on the far left is the side view. In the middle is the top view. As you can see, you can see different spots on the top of RBD regions and NTD regions. On the right highlights one particular antibody we isolated. As you can see, you can recognize the RBD domains. In fact, maintain potent neutralizing activity against many strains that isolated or identified so far. In order to study the impact of those mutations on the neutralization activity of monoclonal antibodies, convalescent serums, or immune serum induced by the vaccines. So the group, including us and then many others in the United States, to study whether these mutations has any impact or have any reduced sensitivity to neutralization. Next, please. So this slides, I borrowed it from Dr. David Ho at the Arizona East Research Center in New York, in Columbia. You can see highlighted the sensitivity of those mutants to the neutralizing activity of convalescent sera on the left. You can see the wild type one in the middle, sensitivity highlighted in black, and then the variants derived from United Kingdom highlighted in purple, B117, and then the orange represent the variants isolated from South Africa, as mentioned by Dale just a minute ago. As you can see, there's a substantial drop in neutralizing activity against South African strains compared to the wild type strains, although the B117 remained relatively similar. In terms of neutralizing activity from immune sera, you can see the B117 remain relatively similar to the wild type, whereas B117 from South Africa dropped substantially, about 7 to 9-fold. Immune sera from Pfizer vaccines induced neutralization activity also dropped substantially. So based on those data and also many data collected from different laboratories worldwide, so it's fair to say, next slide, please, that this result demonstrates the variants has a major antigenic shifts, in particular those identified in South Africa and Brazil. And then we also show those variants has increased their capacity to infect the host, the cell that express other ACE2s derived from different animals. So those changes also really pose a serious challenges to our current antibody therapies and vaccine protection. Thank you.
Thank you so much, Dr. Zhang. That was a great summary. And now we're going to also switch to another time zone. We're welcoming Dr. Misra. I think it's 1:30 in the morning for you as well. So thank you so much for joining us. And we're going to hear about vaccine and diabetes and perspectives from India. Thank you.
Good morning and good afternoon. Thanks, Lata, for giving me this wonderful opportunity. Next, please. I have 6 slides. I'll tell you 6 short stories. All basis of all is vaccine. Now we are undergoing a giant tsunami of the second wave. There are consistently more than 300,000 cases per day for last 3 weeks. Number of active cases and deaths have been staggering. And you can see on the top graph how the second wave is going straight up and nearly all ICUs are full in the major cities. There is a widespread collapse of healthcare with shortage of beds, oxygen, drugs, personnel. Small hospitals are telling patients to come with their own oxygen and remdesivir. You can see on the right side a cartoon. Angry nurse says, you have got bed, oxygen, and remdesivir. Why didn't you bring a doctor? And now COVID-19 is surging in rural areas. Next slide, please. Now, what caused it? And while India was doing so well till January and February, uh, policymakers were totally blindsided by this virus blitzkrieg. Decreased cases in January, February led to general belief that India has defeated corona, and number of crowd gathering events were organized at the same time. You can see cover of time here, a nation overwhelmed. Now this is COVID-19 version 2. Virus appears to be different, very rapid spread. One in the family affected, others are 100% to be affected despite isolation and all precautions. A rapid rise of variants, possibly airborne, possibly more virulent, and likely affecting younger population. Next slide, please. Now, on the left side, you can see election rally in our state of West Bengal. Uh, and next slide, please. And huge, rapid rise of COVID-19, almost 500% after this rally. On the right side is Kumbh, a religious festival which involves people to take a dip in the holy Ganges, was attended by 3.5 million people. It was followed by rapid rise of COVID-19 in all the attendees. Next slide, please. Now, we are the world's largest vaccine maker or vaccine manufacturer, and we have the largest vaccination drive in the world. It started very well with 2 vaccines, Covaxin and COVID Shield. Initial planning was also pretty good. Initially, with healthcare workers, and then above 45 years with comorbidities, and then vaccination was open to all adults from May 1st. Problem started when the cases spiked. People avoided vaccination and vaccination centers. They themselves became COVID hotspot, as you can see on the right side. While India claimed that it attained the largest number of vaccinated people in shortest period of time, a shortage of vaccines are quite visible. Additional vaccine brought in, the Sputnik, and vaccine acquisition from other countries was made easy, but we have a lot of centers, vaccine centers, which are shut now at this point, and total number of population, 2% only has received 2 shots of vaccine. Next slide. Next slide, please. Yeah. So this is a topic which is dear to my heart. Diabetes and COVID-19, a bidirectional epidemic, feed on each other. We earlier did a study which we published where we saw that new onset diabetes during COVID-19 era is far more severe with high blood sugar and high A1C. We already have a lot of diabetic patients in our country, second only to China, and large majority of them are uncontrolled. So huge number of uncontrolled diabetes, over which major spike in uncontrolled diabetes has been seen due to indiscriminate use of steroids. And as you can see on the right side, rising threat of rhinocerebral mucormycosis. A question to be considered is durability of vaccine-induced antibody response, how much it would be, and how long it will last in relatively immunosuppressed, uncontrolled diabetic patients. Next slide. Now, this is very important to discuss breakthrough infections. They have been documented by CDC in our center, diabetic center. Out of 113 fully vaccinated employees, 19 got COVID infection. Overall, breakthrough infections were 13%, and most of them were mild. Neutralization studies with Covaxin or Covishield showed positive result and correlated with mild disease and breakthrough infection. Last slide. And this is again very important, rise of the mutant. B1.617 is mutant of concern worldwide now. This variant carries two important mutations, which are vital for antibody-based neutralization. And the third mutation enables virus to enter cells a little better. This is started with the state of Maharashtra, now all over India. And this mutant escapes inhibition by the monoclonal antibody and to some extent against previous infection and vaccination. And this remains a threat to other nations as well. Thank you.
Thank you so much, Dr. Misra. That was a great summary of the tragedy that's happening in India and hopefully lessons that we can all take moving forward. Our last speaker is Dr. Aruna Subramaniam. She's going to talk about situations in which vaccines may or may not work, specifically immunocompromised hosts. Dr. Subramaniam.
Thank you, Dr. Singh. It's a real honor to be here talking to all of you about COVID vaccines in people who are immunosuppressed. And a lot of them are immunosuppressed because we've given them transplants and we don't want them to reject. So we're giving them anti-rejection medicines. And so we're artificially suppressing their immune system. These are people who need protection from COVID-19 quite badly, and they need the virus to work. Next slide, please. So we know from our cohort studies that people who are immunosuppressed do have poorer outcomes. Our largest cohort study in stem cell transplant patients had 318 recipients and found that a large percent did go on to require ICU level care and 14% required mechanical ventilation. Their 30-day mortality was 33%, quite high. And risk factors for death were older age, if they were within a year of transplant, or if they had lymphoma. On the solid organ side, we have our largest cohort is of 482 recipients, of which 27% required mechanical ventilation for oxygenation. And their 28-day mortality in this study was 19%, still quite high. Their two risk factors for death were age and other comorbidities. So how do we protect these folks? Through vaccination. Next slide, please. Unfortunately, we're finding that the antibody response that they're making, even after 2 doses of the mRNA vaccines, is not as robust as we would like. So this study from Johns Hopkins had 658 participants, all different types of solid organ transplant recipients. Some were healthcare workers, actually. Only 15% developed antibodies after their first dose. They're the people in blue. And they continued to have antibody response after their second dose. The people in yellow are the remainder who never developed a response after their first dose. Their 39% went on to develop antibodies after their second dose. But that left 46% who didn't respond, who didn't develop antibodies after getting both doses of vaccine. So only 54% actually had antibody-related protection after getting 2 doses of an mRNA vaccine. That's a little disappointing. Next slide, please. And who really does not produce antibodies? It's people, of course, who are older with transplants, but the other risk factor for negative serology after both doses are people who are on high-level immunosuppressants, especially if they're on high-dose corticosteroids for the previous 12 months, or if they're on antimetabolites like mycophenolate mofetil. This is something that really inhibits B cell response, and people don't produce antibodies. Next slide. We also have studies showing that people have diminished T cell responses. Thankfully, they have CD4 responses. You can see on the left, the number of people who develop CD4 responses, even in kidney transplant and hemodialysis, are quite high compared to healthy controls. But the bad news is that the percent of actual CD4 cells that are responding to the spike protein are lower, you can see in the second panel, compared to healthy controls. And on the bottom, you see even worse response in terms of CD8 or cytotoxic T cell response. Both the percent of people who respond who are kidney transplant recipients and the overall numbers of CD8 cells that are responsive are diminished in this population. Next slide, please. So our recommendations from our transplant societies take these things into account. We still want to vaccinate all transplant recipients, even if they have limited response. Some protection is better than none in order to prevent or reduce severity of clinical disease. We also really want to give priority to household members and caregivers. It's called ring vaccination, really getting protection to the transplant patient by getting protection to all the people that are surrounding them. Next, we still recommend continued masking and social distancing, regardless of vaccination status in these immunocompromised patients. So the CDC opened up mask wearing if you're vaccinated, but that doesn't really affect our transplant patients' recommendations. We do recommend that they get vaccinated after recovery from COVID-19 to try to further boost their immune responses. The timing is a little tricky. It's preferable if they can get it before transplant, 2 weeks, up to 2 weeks before transplant, so that they will have a robust immune response. If they're post-transplant, we recommend they wait at least 1 month, sometimes up to 3 months, if very severe immunosuppression is used for their induction therapy. We don't recommend routine testing for antibody response, but this may change. We are studying a third dose of vaccine and other strategies to try to boost response in these folks. And right now, we're not recommending routine immunosuppressant adjustment, but this may also change as we get more data. That's the information I'd like to share about immunocompromised hosts.
Thank you so much, Dr. Subramanian. So that's been a great summary from all of our speakers. And I'd like to invite all our speakers to join so we can help do the question answer session. A number of great questions are already coming forward. And I have some as well that I have written out. I thought maybe I would start with a question that I think is probably on the minds of many people, and maybe Dr. Hu or Dr. Zhang can help us answer, which is, will we need a booster and when? And how will we know that boosters for the vaccinations are needed?
Thank you, Dr. Singh. I think the way things are looking, and Dr. Zhang has highlighted that really well, is any virus is under a lot of selective pressure, and they will be generating variants, as we already know. So I think we should be prepared, as the vaccine companies are already doing, as they're preparing modifications to address these variants. And I think it will be very likely that there will be booster shots, very similar to what we do with annual influenza vaccines.
Mm-hmm.
Dr. Zhang may want to comment more.
Yeah, and maybe I could follow up on that and see if Dr. Zhang, do you think that since the mutations are occurring in the spike protein, and that's a lot of sort of where some of the vaccines are targeted, what about thoughts of having sort of a whole recombinant, you know, like a booster that has more of the virus in it? Maybe Dr. Zhang, you could help us think through that. I think you're muted.
Okay, can you hear me? Oh,
Yes.
That is actually various companies are trying to do, including the inactivated form of vaccine that include more protein beyond just a spike protein. Mm-hmm. But based on our immune protective understanding, the antibodies and immune responses, including T cell responses against spike protein, perhaps playing a major role. So while we are thinking about booster, perhaps the immunogen we use will likely focus on the spike protein as well.
Great. There's some questions I see, and also I think that we all have, and maybe Dr. Misra can help us talk about this. what are some strategies to increase the global access for vaccine? One of the our attendees is asking about giving a single dose of vaccine to somebody who's already had an infection, or thinking about other strategies so that the current vaccine supply can go to places like India and Nepal and other places where it's desperately needed.
Let me tell you, India is another story, because we had a lot of vaccines in January and February, and approximately 3.5 crore vials were exported to 94 countries.
Yeah.
So we have had enough vaccines. Only thing is, currently, the supply demand, there is a great amount of mismatch. And because of enhanced supply, as far as India is concerned, the AstraZeneca vaccine export is also limited now to other countries. So actually, global supply of vaccine was affected because of this second wave and a problem in the mismatch in vaccine in India. But there is a great amount of disparity as far as vaccine availability is concerned. And, Indian policymakers and many others have requested US and other countries who have excess vaccine to actually give away those vaccines to other countries. And still, India probably will be able to do something about the current deficiency, but many other countries will not be able to do. And so there maybe in the African countries, in some of the South America, it is much needed at this point of time. And this supply mismatch must be corrected, because after all, not only the current COVID-19, but variants are rising, and unless we have vaccination population, the people will keep on dying. Now, there may be a milder disease, breakthrough disease after vaccination, but death will be there if we don't vaccinate.
Yeah, it's really clear what COVID-19 has shown us, that how interconnected we all are, whether it's about human travel or vaccine spread or variant spread, or even like what you talked about, that the crisis in India is now going to inhibit the vaccine production for the rest of the world. So certainly many tragedies in the making. Can you help, maybe Dr. Hu or Dr. Misra or Dr. Zhang can help us think through the concept of mix and match vaccines? We're hearing a lot about that here in the Bay Area from families and friends who've maybe gotten 1 dose or 2 doses of AstraZeneca. If those individuals end up in places where Pfizer or Moderna or other vaccines are available, what is the thought on sort of variants emerging through some of the vaccinations? What's the thought on mix and match possibilities?
I might try to take a first stab. In general, when people use the vaccine, where there's a 2-dose, there's a concept of a prime, where it kind of primes the immune system followed by a boost. And a number of different studies have shown when there are different types of prime boost, or they call this heterologous prime boost regimens, that it induces a broader immune response. And due to the differential availability of different vaccines, not only the public, but a number of different researchers around the world have said, well, let's look at what might be the effect on the immune system with different heterologous prime boost regimens. So that clearly is an active area of study. And I think, it's certainly worth investigating, because it has been shown to be helpful with other infectious pathogens. And I'm sure the others have additional comments or thoughts.
Yes.
Sorry.
Go ahead, Dr. Misra.
Okay, I can refer to a small study which has been published yesterday in Lancet, that they mixed 2 vaccines, and that is AstraZeneca and Pfizer. And the main outcome that they saw was adverse effects. Adverse effects were significantly more once they were mixed, and that included fever, fatigue, headache, etc. So I can tell you that the adverse effect will be more. Efficacy, you have already answered.
Dr. Zhang?
Yeah, I would agree that this is a practical and scientifically reasonable idea. And then based on the publication just Dr. Misra mentioned, they have shown some boosting effect measured by various of assays. So give us some hope that the mix and matches would happen. But I have to stress that regardless of what strategy, this has to be carefully evaluated, perhaps in the clinical trial settings or real world settings. So the mix match could be maximized and the safety could be protected.
Yeah, I think those are really important points. We are lucky to have multiple vaccines across the world that are safe and effective. And we want to sort of keep that going, because I do worry that otherwise, if we have adverse events or other issues, that even more vaccine hesitancy will develop. I thought, Dr. Subramanian, I might talk to you about your patients, immunosuppressed patients who may not respond as well. Can you talk to us a little bit about other approaches, maybe monoclonal antibody treatments that could be sort of an adjuvant to vaccinations, maybe even pre-exposure prophylaxis, for example?
Right, that's where, I feel like our immunosuppressed patients need extra protection. And so monoclonal antibody use, even for treatment, even convalescent plasma has been used more in these patients in treatment settings, but as pre-exposure prophylaxis, that, it is being looked into in the first few months post-transplant, as even monthly infusions are being thought about, especially if there are household contacts. It's maybe beneficial to give them monoclonal antibodies early. Unfortunately, monoclonal antibodies are not available worldwide in many settings. So this is a strategy that we have the advantage of looking at in the US, but unfortunately, it's not something that can always be used worldwide. But that's really a way we're looking at. And even the prime boost vaccine strategy is being thought about in transplant patients as well as maybe a third dose. So there are various things we're thinking about in our immunosuppressed patients to give them some extra protection.
That's great, thank you. I thought maybe I could ask each of you to try to dispel one common misconception that you've heard about COVID vaccines, either from patients, colleagues, friends. What are things you want to say to the people listening that they can help take that word forward? Maybe Dr. Hu, start with you.
Thank you. Well, before I mention that, I did want to add to what Dr. Subramanian mentioned about antibodies and vaccines, because right now, currently, there are a number of companies that are actually partnering together to look at whether receipt of an antibody might possibly adversely affect the immune response from a vaccine. Now, the CDC's recommendation was really based not necessarily on scientific evidence, but the observation that that reinfection didn't occur over a certain period. But now I think a lot of companies are working together. For instance, Regeneron and Moderna are looking at things. Vir GSK and Janssen are also looking at that combination. So I just want to add that I think one of the things that has been beneficial about the COVID response is there's been a lot more collaboration between companies and academic researchers. So I think one thing is not so much a misconception, but I think one thing that has also been fortunate is prior to this epidemic or pandemic, I don't think the general public really understood about clinical research, phase 1, phase 2, and phase 3 trials. And now I have people like neighbors, and they'll come out and they'll say, now that the EUA is here, when will the FDA provide approval? And I think the level of sophistication, certainly in the United States, but I'm sure internationally, has really increased. And I think that that's really important. And the second thing I think to really point out, and I mentioned earlier in my talk, is that whenever you give vaccines, there will be adverse events that are related to the vaccine. There are ones that will occur that are not related. And it's important to look at the baseline level of whatever event is under question. And even if there is something that is related, I think it's really important to balance what the risk of those events are, which in this case are very, very rare, with the incredible protection that all these vaccines are showing against severe disease and death. Thank you.
I remember seeing the data come out from Pfizer, and the efficacy data. You get goosebumps, right? The first time you see that, because I've never seen a curve like that. So I think, yeah, such a powerful moment for science and for industry and academia and patients and NIH working together. So that was great. Dr. Zhang, what are your perspectives? What are things that we want to emphasize to the listeners of this meeting so they can take forward to friends and colleagues and patients?
Yeah, despite of the great success and progress we made in such a short period of time, I think there's still many misconceptions and people around me that are still thinking, in fact, many of them thinking that vaccine will cause more harm than good. So how, as a scientists and clinicians and vaccine developers, to disseminate the right and the scientific information to the general public is still one of the greatest challenges that we have to overcome. So for the audience, that I want to say, from a scientific perspective, and vaccine has been very safe, has been very effective, and has gone through a very thorough process of evaluations. So we should remain confident that in order to stop the pandemic, the vaccine is such an important weapon. We all have to capitalize on it and disseminate the right and scientific information.
Yeah, I think those are crucial points. Dr. Misra, what perspectives or thoughts would you like to share?
Yeah, I see a lot of people from India who have logged in, and there are a couple of questions also from India. Long questions have been written. So I want to give a couple of advice to Indian listeners. There is a great divide about vaccine hesitancy and perceptions about vaccine between the upper socioeconomic strata and middle and lower socioeconomic strata in India. And currently, the epidemic is moving towards the low socioeconomic strata, towards the villages, and towards the urban underprivileged. Now, there people are not vaccinated. And there are a number of issues that they have vaccine hesitancy. I don't want to name them, it will take a long time, but essentially they are highly hesitant to take any vaccination. Now, all of us who are relatively privileged in India have a duty to do, and that is to tell these people, underprivileged people, to actually get vaccinated and tell them in simple words that ultimately it will prevent your health from deteriorating or even death from occurring in you and your family members. So, for example, I have two maids who come and they never took vaccine, and we told them, and then after that they took vaccine. And something like this must be done by everybody who has some connection to underprivileged people or poor people.
I think that's a really important comment for the world, actually, not just India. We all are privileged in terms of our education and our access to science and things we just take for granted that not everybody does. I had a patient who insisted that the mRNA vaccine was gene therapy and that it was going to change his being. After many conversations, he got vaccinated, and he's still the same, surprisingly. But I think those are surprising things. Dr. Subramaniam, what's your perspective, both from your patients as well as your connection to India and other places?
Yeah, for our immunocompromised patients, yeah, I've heard the same thing. Does the vaccine is it going to change your DNA? And that's something that you hear a lot. And in our transplant patients, it's been drilled in them that they shouldn't take live virus vaccines, and they hear adenoviral vector. In some cases, they get very nervous that, oh, is this a live virus? And so sort of dispelling that myth. No, it's replication incompetent. It can't make more copies of itself in you. You're not, and some of them have had adenoviral infection and they're scared. So those types of things really trying to counsel people on the safety of these vaccines, even in immunocompromised patients. And, that it's not, that some protection, as I said before, is better than none. Some people are also getting very depressed when they read these, and why should I even get vaccinated? It's not going to help me. But 54% did develop antibody response after 2 doses. So, and that CD4 response that people make, it can be quite helpful. And so I also have to give people a pep talk that this is really going to give protection, and really getting their family also. Sometimes family members are willing to do anything for their loved one, and yet they don't think that the vaccine is an important thing for them. So that's another thing that we really emphasize for our transplant patients.
Yeah, that's really important points. I thought maybe I would ask Dr. Hu and Dr. Zhang to talk about this mRNA technology has been a game changer. Tell us a little bit about whether you think this is going to be a game changer for most vaccinations moving forward, and how has this helped us sort of be prepared for the next pandemic, which we know will happen, hopefully never at this scale, but we know that emerging viruses and emerging infections will happen.
Yeah, the mRNA approach actually isn't just started with this vaccine. There were a lot of research with other vaccines using this approach. But I think the main takeaway, and this might be a little bit too technical, but in the past, people were concerned about RNA in particular because there's a lot of enzymes in the body, RNases, that break down RNA. But due to a lot of technology in terms of modifying the RNA, delivering them in different manners, such as lipid nanoparticles, they've really kind of optimized this approach. And the real advantage is that these can be kind of almost like a cassette, plug and play. You can replace the immunogen of interest, and these can be manufactured relatively easily compared to some of the traditional vaccine production approaches. So I think with the new variants, as well as new potential pathogens, this type of technology will be useful. I did also see in the chat that people were asking about different delivery methods, such as nasal inhalation. And like the flu vaccine, I think a number of manufacturers, not only for active vaccination, but also for antibodies, they're looking at a number of ways of delivering, not just through intramuscular injection, but subcutaneous, intranasal. And all of these things, I think, go back to the importance of supporting both basic and clinical and translational research.
And having the infrastructure to sort of implement right through these large scale studies. Dr. Zhang, what about its implications, and as well as maybe for HIV, something you've studied for a long time?
Yes, in fact, the mRNA technology has started HIV vaccine efforts years before the COVID-19 outbreak. I agree with Dr. Hu that the mRNA technology is one of the plug and play platform that allowing us to react very quickly, effectively. So given its speed, flexibility, and precision for viral antigens, I'm sure this technology will have a great future. However, I have to say that this is a relative new technology. It has just approved for COVID-19. There's much room for improvement, and not just for the antigen design, the delivery technology, and all the storage and deliveries. So the scientists and the industries in the field has been working tirelessly to improve those shortcomings. In the future, we are very confident that this technology will offer unprecedented speed, flexibility, and precision to tackle with all kinds of variants and perhaps unknown pathogen in the future. So a very bright future.
That's great, thank you. I want to leave a few minutes to see if people on this panel have questions for each other.
I have a question for Misra about the current epidemic in India. How much of those infected are caused by the new variants, or how much of those are caused by the public health measures or ineffective public health measures? Yes.
Yeah, it's difficult to say which one is more responsible, but I would say collectively all factors are responsible, and the rise, whenever it was rising, the markets were totally open. People were all mingling around, and there were gatherings. As I showed you the photographs of those gatherings, these are just two gatherings. There were many more. After all, election rallies are held at 50 different places, and each one has at least 10 to 20,000 people, and many of them without masks. So this was one thing. Opening of markets was second thing. At the same time, perhaps the rise of the mutants, and especially B1.617, is something which is rapidly spread. And Maharashtra, which was in terrible state around 3 weeks back, this is the large state in the Western India. This mutant was responsible for almost 30 to 40% of the spread. And now this has spread to various other states, and it is also mutant of concern in UK. Also, maybe it will spread to other places too. So I think all this and lowering of our guard, policymakers lowered their guard totally because they thought that everything is over, and we have won, and we have hit the home run. That was the feeling in January, February.
Thank you.
Maybe I'll end with a provocative question for Dr. Hu or Dr. Subramaniam. What do you think about the new CDC guidance that has come out that we can throw away our masks if we're vaccinated and not traveling on a plane?
I actually am glad I've retired from the public health service. I might be able to be more. It's a complex question because one has to balance kind of. I think CDC was put in a difficult position because on one hand they want to encourage people to return to life as normal, and I think they want to encourage people to get vaccinated. So this was like an incentive. If you get vaccinated, you can return to normal. I think the only caution that everyone has mentioned on this panel is that the virus is very tricky, so to speak. It's always evolving to kind of combat whatever preventive measures that we try to put in place. And I'm actually really worried that come this summer, come this fall, there will be new variants, not just the ones that people have been concerned about, that may have another wave. And as Dr. Misra mentioned, there are a number of countries that initially were very confident that their measures were very effective, only to find that later spread of different variants have occurred. So I think on one hand, it is good news that there's more people vaccinated, but I do think we always have to be cautious.
I think that's a perfect way to sort of end our session with hope, and joy that the vaccines have brought us, but with a little bit of caution that the virus does seem to be 5 steps ahead of us. And we want to be careful that we don't get caught flat-footed, unfortunately. So I just want to end the session by thanking the phenomenal speakers. Thank you, Dr. Subramaniam and Dr. Hu, and a special thanks to our night owls, Dr. Misra and Dr. Zhang, for joining us in the middle of the night. I think this has been great, very informative. And then this is a perfect segue to our next session, which is going to be on mental health. And I don't know about you guys, but I need some help in this area after this last year. So it's my pleasure to introduce everyone to Dr. Rona Hu, who's a Stanford professor of psychiatry, who's going to lead this session. Please join me in welcoming her.